We are fortunate enough to be joined today by Dean Christopher Yip to deliver this mock lecture. Professor Yip is Dean of the Faculty of Applied Science and Engineering. He is also a leading scholar in the field of single molecule biophysics and a faculty member with the Department of Chemical Engineering and Applied Chemistry, the Department of Biochemistry, and the Institute of Biomedical Engineering. He is a principal investigator with the Donnelly Center for Cellular and Biomolecular Research at the University of Toronto. He has provided leadership to more than 100 research engineers and scientists engaged in discovery and product development in the areas of neuroscience and sensory stimulation, biomaterials and tissue engineering, molecular systems biology and nanotechnology, as well as medical device and drug delivery system design. He is the recipient of several prestigious awards for both his research and teaching at U of T, in addition to being named a Fellow of the American Association for the Advancement of Science, a Fellow of the Engineering Institute of Canada, and held a Tier 2 Canada Research Chair in Molecular Imaging. You can also continue the conversation today with Dean Yip by using the hashtag #CoffeeWithChris and following him on social media at UofT Eng Dean. So Dean Yip, over to you.
Great. Thanks, Josh. And thanks for that intro. It sounds like I do a lot. And today's mock lecture is going to focus a lot on actually what my lab does. I will do a shout out for U of T Engineering and my mug and the Coffee with Chris side. So for those who do come to U of T, feel free to reach out, and I look forward to chatting with everybody. So today's mock lecture, it's going to be a little bit about what I do in my lab and that unique interface between engineering, biology, a little bit of other stuff, and show you some of the stuff that we're doing to answer some really cool questions. And some of them are a little bit timely. So I'm going to jump to actually my first slide. And kind of to pick a timely topic, asking ourselves some interesting questions. I don't know, how does the virus get into your cell and what can you do about it? And how can we prevent it? How can we understand what's going on? How does it happen? How do we prevent it ultimately? And what's it trying to do? What's the cell trying to do? And you know, how are these things sort of working together? And I titled the talk, Mapping the Molecular Makerspace, because it's all about how things are coming together to do their function, right? Virus wants to get into your cell probably trying to prevent it from getting the cell, but somehow the virus figures out to hijack it. We as scientists and engineers and researchers are trying to develop tools and vaccines and things like that, which would prevent the virus from getting into the cell, or once it's in the cell, preventing it from reproducing. So how do we do that? And really try to understand the kind of the intricacies of really what's going on. And so for my lab, you know, we're really interested in, you know, what's the best way to kind of understand how things kind of come together, right? And for us, it's all about kind of like, well, can we watch it? Can we see it? Can we measure it? Can we characterize? It's kind of like a classic engineering challenge, right? And I'm going to kind of put up a slide which sort of shows sort of the scale of which things are happening, and you can see kind of on the far right, in a sense, you know, virus is like 100 nanometers, right? And this sort of sense of scale, if you happen to see a mouse in your house, it's about 10 centimeters long. And you can see that with your eye. Some of you may have had an MRI or an ultrasound at some point. You had an injury and you kind of get a sense of the scale, cells, bacteria, and so on. But you know, here are the tools we've got here, and I've got the slide sort of showing you. And you know, we want to see things as they happen. And, you know, light microscopy is probably one of the best ways to do it. But I said earlier, I want to kind of watch how things happen. And I use a slide of the virus when I come in. But there's this gap here. And so a question becomes, how do we probe and break that gap? How do we kind of come down on that scale and try to understand things? And you may go, well, there's all sorts of new technologies coming out. I did put electron microscopy because that's a really powerful tool, but it doesn't work well for things that are alive. And then you sort of go, well, you know, I'm sure you've all used a light microscope at some point. In fact, I kind of did a long time ago. And you know, here's a picture of me, just to set the scale. That's me on the left-hand side there. when I was a little guy looking down a microscope in a lab, it's a classic picture. And then me looking down a microscope a few years later, I was at the university, but nothing's really changed. And so we're intrigued by this sort of like, how can we make it better? How can we use ways of imaging things to see features that are small, see features that are dynamic, and try to understand what's going on? And so I'm motivated a little bit by this idea of visualizing things. So I'm gonna kind of give a nice bit of a construct. I like using this one because it works well. So this is a kind of Google map, right? That's Canada. That's Toronto. And some of you are in Toronto now, some of you maybe around elsewhere in the world. But on a Google map, right, just like on a microscope, you can zoom in, right? I can keep zooming into Toronto, right? There's, oh wait, there's campus. I kind of wish we were actually on campus right now. I know a lot of us wish we were kind of in person giving this lecture. I can keep zooming in. And what's interesting here, this is a picture, this is a schematic of campus, or outlines the buildings. But if you take a satellite picture, you get a 3D view of the campus, right? And I'm keeping the zoom in. I'm actually going to zoom in on the building where my lab actually is. And I can keep going, right? It's a little bit interesting, the fact that the satellite can keep zooming all the way down like this, so all the way down to College Street. And then you kind of go to a view that I'm sure, almost positive, everybody has done at one point, right? You've all done this, right? You've all street viewed your house, your friend's house. You definitely have street views, like, I'm going to go to this restaurant. Where is it? What does the building look like? And you get on street view and you kind of like, there it is. That's what it looks like. And I'm sure actually maybe some of you, in fact, jumped up in front of the Google Street View cars that's been driving around taking pictures. And so this is cool. Like, this is a picture. It's good. It's a picture in front of my building. A couple of people walking around. Kind of classic, they're sort of stitched together and so on like that. But imagine if this was a picture of a cell, right? And I'm zooming in. And these people here are actually proteins or molecules moving around in the cell. And you take this picture like Street View does. You all know that the Street View picture of your house, the restaurant, wherever, wasn't taken today. It's not an instantaneous picture. It's also not a live picture, right? And if you look at this picture closely, you can ask her a bunch of different questions here, right? The two people in the center of the frame there, I could ask a question: do they know each other? Or, as the picture was taken, do they just happen to be walking by each other, right? Where are they going? I don't know who they are. I just know that there's two people walking through. The other two people, they're walking. It's a stop action picture. It looks like one person, I think, is one person's walking to the left and one person walking to the right. That's okay. But there's an interesting thing in this picture that you have to look closely at this. Again, it's easier when I'm in person, but what do you notice here? This person appears to have 4 feet. So I'm kind of confused. Normally people don't have 4 feet. So the question becomes, what happened that gave rise to that picture? And if this is a biological question or in a cell, it's like, well, what if this is a protein, what's it doing? Is it coming apart? Is it separating? Is it splitting? Are they moving in different directions? And so we're interested in not just stop action pictures, but live action pictures. We want to understand what's happening here. Maybe it's, this is a schematic, but is it two people coming together to form one? Was it two images collected together? How did this individual suddenly have 4 feet, right? And so we're really, really intrigued by following things in real time, right? So here's an image, and this is a live image, well, recorded image of proteins moving around in a cell. And I hope you can see it on your screen, but you see there's some things which look kind of static. And this is a picture of an entire cell, and you're kind of looking at the bottom of the cell here. And you can see stuff kind of shooting all over the place. Some things are wiggling. If you look closely, some things seem to move, stop and start. They go left to right. Some of them seem to kind of roll around in circles. Some seem to be like running on railroad tracks. It's all the same protein that we're looking at. It's the same molecule, but where is it Why is it there? Where's it going? That's another key question. And like, what's it doing? What's the connection between all these things, right? And if we look even more closely, we can follow intriguing things here, right? So watch that kind of starburst. I mean, it looks like something exploded. And what's intriguing here is this is actually, you're watching molecules, proteins inside a cell, come up to the cell surface and then merge with the cell membrane, which is the outside of the cell. And so how do they know where to go? How many molecules are there? How fast are they moving? What's telling them to go where? Like, what's that roadmap? I don't think they have a Google map that says turn left, turn right, and walk 50 feet and go here and then explode. Why is that happening? What's the driving force? If I flip this problem the other way around, it'd be the same as watching a virus come onto a cell surface, land, and then somehow merge into that cell membrane. So watching these things live is such an powerful tool to understand what's going on. So this is cool because these are actually really rare events to watch this sort of starburst effect. But if we watched another series, you see these sort of cool little wormy guys, folks kind of running around. It's like, what are you doing? Why are you splitting, right? How many molecules are there? Where are you going? What is happening at these particular points in time? What are the factors driving this? And so we're intrigued by, can we develop strategies to answer these kind of questions, right? And again, we're using tools which have been around forever, but how do we enhance their capabilities and their performance? That's really kind of the stuff that we're interested in, because we can really start to answer some really, really important questions about interactions, dynamics, what's going on, where things are docking, where and stuff is happening. So here's kind of the motivation of what we're trying to do in my lab. We're trying to build tools that let us look on time scales from the nanosecond, millisecond to days, on length scales from single molecules all the way up to centimeters. So remember that length scale I showed you? And we want to follow events dynamically as they're happening, okay? And continue to image, because it gives us a lot of insights. Now, the image in the upper, on the, I guess, right-hand side here, it looks weird. It's like 6 images and something seems to be wiggling around and stuff on. And what I've below is what's actually happening. What you're watching is a fruit fly developing in real time. And you're seeing the cells which make up that fruit fly start to move, start to change shape, start to interact, and start to form structures inside the fruit fly as it's growing. So it's going from sort of cells in just a giant bag of cells that don't know what they're going to do, to forming different structures like their heart, the eyes, the brain, the stomach, and so on. And so we now have these tools that let us follow this as it's happening, as that organism, as that animal is developing in real time. And that gives us a huge opportunity to understand what's going on, how we can interact with that process, what could happen if things, for instance, get exposed to different chemicals in the environment. How does that change the process at which these take place? In the lower left, I'm going to show you a little bit later, is how we're breaking some of the fundamental laws of physics. Laws are there to be broken sometimes, to give us an enhanced resolution. I'm going to explain a little bit later how we go from square pixels to little round things. We're going to motivate this by a little bit of a collaboration I have with a colleague, Rodrigo Fernandez-Gonzalez, another professor in biomedical engineering. Interesting background. He's an undergraduate in computer science, but he did his PhD in cell biology, and he's now in biomedical engineering. So there's an individual who sort of moved around and ended up in engineering as a faculty member. But Rodrigo, aside from being an absolutely stellar marathoner and someone I'm trying to keep up with when I run marathons with him, is really interested in the forces involved in how wounds heal, like when you get a scratch, how do the cells know where to go? How much force can they exert as they do that? And what happens during development? And so he's intrigued by the question of how do you measure the force in a cell, right? You can go to the gym right now and grab a weight and lift it, and you can measure the force that you're doing. You can measure how much force you're exerting as you're running, for instance, but this is a cell, it's small. And so how do I I can't put it in the gym. I can't like grab it and pull on it actually very easily. It's kind of difficult. So how do I do that, especially as something's growing, right? It's like the previous picture, like the cells are moving around but they're changing shape. How do I measure those forces? And so Rodrigo, outstanding scientist, outstanding computational scientist, he realized that if I look at the shape of the cell, I can actually calculate the force, right? The forces in a cell are different if it's a sphere or a circle versus it's an oval or some other shape. But then he said, well, you need to give me a tool where I can follow this, right? So we said, well, let's try to build you a microscope that can actually do this. So as an engineer coming at it, we said, sure, there you go. We can do that. We can take lasers. Lasers are always fun to play with. Some mirrors, a couple of optics, a camera, and we can build you a microscope that would allow you to now watch how cells grow and develop in these particular systems. So as an example, here's a case. Here we're watching two different sides of that fruit fly embryo as it's growing. Now, the clock here is in minutes and then seconds. So you see how we're capturing images probably every 2 minutes. You know, that's an hour after an hour. And look at what's happening to this. We're not doing anything. This is just the egg on its own. Those cells are moving around. They're getting chemical cues. They're deciding, I'm going to twist, I'm going to turn, I'm going to form all these cool structures. And we can follow that process in real time. We can calculate now the forces that are happening inside the cells. So what's particularly cool is that I kind of lied to you. I said cells are round or oval, but look what we see here when we take these high resolution images. We in fact see these cells are, wow, they're actually not round. They're like hexagonal. And this is because of the architecture as they're starting to form those shapes, as they're starting to integrate with each other. And from those points of contact and those angles that you measure and the length of those edges, and we're labeling here the outside of the cell, we can now calculate how much force is being exerted on those cells by the other cells in response to those dynamic events which are happening around it at the individual cell level. And you're doing this in real time, which is amazing when it comes to thinking about what the role of force is as something's developing. How would this look different, for instance, if it was developing in space where there's no gravity, there's no gravitational force being applied to it? What happens in a different environment when forces are being applied, and how does it change in way the organism kind of responds to those interactions? So that's one area where we're interested in looking at these kind of forces. But this is big, right? I mean, well, not big, 50 microns, right? It's still small, but it's still within the realm of what we can actually watch with a conventional microscope. So let me get smaller on you and ask a nano question now. And that is, you may remember a little bit from physics or high school if you were taught yet so far, that there's this limit known as the Abbe limit, and that is that optical imaging using visible light is limited in terms of what it can see, about the wavelength of light divided by 2. Blue light, shortest wavelength we can see, at least with our eyeballs, about 400 nanometers, which means at best you can see 200 nanometers or resolve something 200 nanometers in size using an optical microscope. But I think I said earlier as we began to talk that proteins, viruses, all those things are well below that. So we would never be able to see them using a light microscope because they're way too small. Now we know we can see them using electrons, but electrons and X-rays and things that means I got to kill it, right? I can't watch this as it's happening in real time because it's the wrong condition. And so this is a huge engineering challenge and one which has vexed people for hundreds of years, right? Is how do we break this law? How do we break what we were all taught in physics was something you can't do? And so some really kind of innovative folks thought, well, what if we looked at the problem in a different way? And so I'm going to kind of take a bit of a movie here, and you'll see what's going on here. So on the left, that's actually, well, Actually, if we were able to have a conversation right now, I would ask the question, what are we looking at? And anyone who happens to be in France would probably go, well, that's the Eiffel Tower. And most people would recognize that as the Eiffel Tower. But what's intriguing is if you took pictures really fast and simply looked at lights that blink, because they're all blinking at different rates, you could take those blinks and regenerate the structure of the Eiffel Tower purely by how the light is blinking and not by taking a single picture, all right? So in the context of a molecule or a cell, if the light blinks or blinks really fast, or we make it blink, maybe we can resolve it better than if it was on all the time. This is a little bit of a trick. We play with statistics to do this. And so I'm going to motivate it again by an example of how we're able to do this, right? We somehow fake the molecules into blinking now instead of staying on all the time. And so here's an interesting protein. As the title says at the bottom, it's the fourth component of cytosol. This is a really important protein that helps keep cells together, keep their shape, help things move. They actually act like a little bit of a train track for stuff to move around in a cell. And for a long time, people have wondered, like, how do these things organize? And what happens if it's not organized well? If you've got a molecule that forms a ring or a strand, and it's meant to keep your cells intact with a certain shape, and it doesn't work, well, your cells aren't going to behave well. They're not going to be rigid enough. They're not going to strengthen. You're going to have all sorts of problems. But how do we see this? And can we see this in a living system? Can we visualize this? And also, what is the actual architecture? This is a huge challenge for folks, right? And for years, people have asked this particular question. So we sort of looked at it and said, well, I wonder if we could use this little trick of making them blink as a strategy to see what actually happens and how this architecture is actually built, right? And so I'm going to show you exactly what we do. On the left is a standard picture you take of these molecules. And sure, if you stand way back, it kind of looks like a ring, right? It's like a really blurry ring. But when you zoom in, right, just as if you were zooming in on a picture you took with your cell phone, it's all pixelated, right? And that's the best we can do. And remember, proteins are small, right? They are, 20 nanometers or even smaller. And so what we do is we take blinky pictures, right? We take pictures as the molecules are blinking, and we keep building them up. And you can see on the right-hand image, as it goes, you can see how it starts to blink, and you start to create a new image. You just keep adding these things together, and you create an image which is about 10, 50 nanometer spatial resolution now, well below what we would have normally been able to see with the classic microscope. And so that image that's all pixelated, when we use this other technique, which we've helped work on, we now have an image which has a much finer detail of where the proteins are that form a ring. And in fact, wow. Okay, so artistic license here, making something look like a ring, because now you're in a cell. Remember, we're actually looking at the molecules in the cell. They're not taken out. They're not artificial. It's in a living system. They're blinking, and we've got the architecture, right? It doesn't look like a perfect circle because the cell is twisted and things are distorted, and you're looking at it on a different angle. So this is really cool. We now have this ability to look at this really neat architecture. We can now see what this structure looks like. But what's even more important or intriguing now is that, remember, this was set up as whoops, let me just go back here for a sec, as not the tower, as different proteins. So what's connected to what? How do we differentiate those? So what we've been able to do, and I'll just kind of step through this fast, is do two colors now. So we do the blinkiness, but we make them blink, make each molecule, different molecules blink. One molecule blinks red, one molecule blinks green, and we reconstruct and we put those back together. And we can now see the architecture of these rings. And you can see how they're twisted, they're in different orientations, but we can now see that the red ones are next to the green ones. I apologize for the color scale here because it's not color safe for anyone who happens to be sort of red-green color deficient. So to you, it may actually look like one color right now, but we can actually now start to see those architectures and how they're arranged next to each other. Super important now because you're doing this in a living system, and the ability to map these architectures, characterize what was previously really just a thought about how they're built up. So this is a really quick kind of overview of sort of the stuff we're doing in our lab around this idea of how you combine optical imaging to address really complex biological problems, whether they're on the scale of watching an organ develop. I use the hint of sort of watching how the heart forms to understand what is involved and the forces involved, all the way down to watching a virus or watching molecules come together to form structures on the cell surface of a cell. So what's next for us? Well, always push the limits of space and time. That sounds kind of fanciful, but we want to image faster. We want to image with higher resolution because there's all sorts of questions. This is sort of one of these sort of endless rabbit holes of questions you can go down because you now have the ability to do this and answer these questions. More colors, different systems, more complex problems. And can we have the computer help us, right? Can the machine and the computer be looking at those structures as they're forming and say, oh, wait, it looks like the heart is starting to form. It looks like something's about to dock to the surface now. Time to change the experiment. Imagine if you're sort of following the development of an organ, and you say, well, what happens if this environment changes all of a sudden? What happens if a drug is introduced at this point? What happens if a different chemical is introduced in the environment? What changes? And so for the microscope to know that it's time to add this versus a student, how do you do that? And how can the microscope be smart? And we actually think there's some intriguing approaches to apply here, which we could actually take, this is from an engineering angle, take from the people who are working on self-driving cars. If you think about it, an autonomous car needs to know whether it's a stop sign, whether it's a traffic light, whether it's a person, whether it's a bicycle, whether it's another car, and it knows from the shape and the different perspectives. Can we train a microscope to know that the heart's forming, to know that an organ is forming here, or that a protein is coming to the surface? Can we train them to understand that, right? So in addition to training the microscopes, my lab is focused on training the next generation of researchers and scientists and engineers. And so that microscope, which I used when I was 3 years old, is currently in my lab, tells you how durable these things actually are, and has now been test-driven and successfully test-driven by my little munchkins when they were 2 and 3 years old. So things keep cycling around over time. But thank you so much for taking a chance to be here for this mock lecture. I'm going to thank as well the sources that sort of help fund this research, but help drive the programs going on in my lab, whether it's the granting councils, the university itself, various other organizations. But I certainly thank you for your time, and I appreciate the opportunity to give a little bit of a mock lecture on the interface between engineering and biology and some of the fun stuff that we do in our lab here at the University of Toronto. So I'm going to stop my share there and happy to take questions.
Fantastic. Thanks so much, Professor Yip. I've got to be honest, I come from a history background, and what I love about a lecture like this is you make it accessible for people, I think, of all backgrounds. I walked away with that, having been able to follow your train of thought, and I think that's really a comment on the strength of a professor.